A very good afternoon, everybody, and a warm welcome on this cold winter afternoon. It is our great honor to welcome you all to the M.K. Bhan Memorial Lecture, a tribute to the remarkable legacy of Professor Maharaj Kishan Bhan. Professor Bhan was a visionary scientist, a passionate advocate for public health, and a dedicated mentor to countless young minds. His groundbreaking work in the pediatric research, vaccine development, and also global health policies that he was involved in has left an incredible mark on our understanding of the human health as well as disease control. This memorial lecture series has been established in NAAB to commemorate this and also to perpetuate Professor Bhan's vision, values, and ideals, and also the commitment that we have towards advancing our scientific temper as well as scientific research. We are privileged to have along with us today two esteemed guest speakers, Professor Sudhanshu Vrati and Dr. Krishna Ella, and Professor Redana, former Foundation Director of NAB here today. I now request our Director, Madam, along with our guest, to honor and offer tribute to Professor Bhan. May I now request all of us to offer a silence for Professor Bhan's in memory of him. Thank you, everybody. I now request our Director, Madam, to please deliver the welcome address.
Thank you, Dr. Madhuri. Very good afternoon to one and all. In the memory of late Professor M.K. Bhan, the Institute has decided to remember him by sharing the wisdom, thought, and enrichment which is there amongst all of us through Professor Bhan's work. This is a second lecture in the series, 7th December. Every year we decided that we will be doing it a little bit with the convenience of a week this side and that side. So with that, I on my own behalf and on behalf of NIP fraternity, thank very sincerely both the speakers who have worked closely with Professor Bhan and in an area of his choice, which was so dear to him. and till end he has been working towards it. So I will just take 5 to 7 minutes and go with a small presentation, which is, if someone want to help me and get the presentation screen? I forgot my specs there. That's my. Put it and show and I can help myself. Thank you. Is it required? Is it required? YouTube Live. Yeah. Put it, show. Thank you. Thank you, Akash. The one of the blessed institute of DBT works with a very specific motto that is human welfare and through animal health. And to set up the tone in a memory of esteemed former Secretary DBT, Dr. M.K. Bhan, this institute is his brainchild, and we all owe a lot to him. I'm also very thankful that the founder director of the institute, Professor Edana, is amongst us. Thank you very much for joining. Professor Bhan conceptualized it somewhere in 2007-8. A journey was not simple for him to bring this institute to the level what it is today and how he has traveled. So this was a journey and started exclusively with the idea, one small idea, but great idea. And this is what today, standing tall in an area of animal biotechnology, the only institute in the country which is working in for animal biotechnology. And to add a feather to its cap, when BRIC came to an existence, as we know, 10th November 2023 was BRIC Foundation Day. So all these things which the institute has been doing is continue to do. And I will just, I think, is someone going to introduce Professor Bhan more? Yeah, so I'll just do that. People know him, but for youngers who know, by profession, he was a medical doctor and he has studied at AFMC Pune. And then he was basically a pediatrician, a noted pediatrician. This is what the rotavirus vaccine. And we have two worthy speakers, Professor Sudhanshu Brati, as well as Dr. Krishna Ella. And I'll leave it here and they would be highlighting and talking more. These are some of the initial days when we were coming up, the NIB. And I'll quickly go through, it was the university and then later Mayapur and then this campus. And it was all happening with such a scientific fragrance that no stone was left unturned and the building thing was being planned and it was coming up. So to make a point, it was the university and then they moved to another rented building, but everything was happening, including good science and bringing the right people at right forum for brainstorm, for workshop, as well as the Nobel laureates who visited this institute when it was building its initial building bricks. This was the site and university and Professor Redana and Professor Syed Hussain. It was all, and this is all what NIB was earlier, 2012, 2014, between that time. So 200 acres were decided and eventually 100 acres. So these are some of the glimpses of Mayapur interim facility. This is the main campus. Dr. Subir is not amongst us, but the land, was given to this institute when Professor Redana was doing, and all the architectural work and all. But surely, all the concrete structure and things, all this is placed we shall always owe it to former director, Dr. Subir Majumdar, and it was happening during that time. And it beautifully developed, not only aesthetically, but inside as well through the science, which I'll take little bit. And it was 11th of August 2018 when this particular campus was inaugurated by the then minister, Dr. Harshvardhan. And then journey continued, and the baton which was passed on to me as a third director. So we decided in last year that uh, in the memory of the person who gave us the institute, there should be some professional discussions, and that science must be celebrated. And so we started. So this is what was the first lecture. We have had Professor Balram Bhargav, and this is all the first lecture. The institute, as I said, has been moving. Just first one, that how scientists interact. So the idea, the Bhan site to bench, what are the requirements? And these are all academically, how much we are connected with students in and across the city, within the country, as the academic handholding. And these are some of the glimpses. I think I'll not take more of a time, but before I come and pinpoint what all the science is going on over here, and we continued, and our students and fellows, especially some of the senior ones who were with NIAB since beginning, they have worked very hard, and I shall fail in my duty if I'll not quote those students and fellows who worked exactly in the same mode, the way scientists and our administrative people were working to bring this institute the place where it is. So, as I said, BRIC. So, we have a Bio E3 policy, which has been announced by Union Cabinet. And this institute is very much in sync. I mean, on the other hand, if large animals are not bioincubator, then what else? So, in fact, it's a best boon to the institute that we are working towards this policy. These are some of the technology transfers, MOU with academia, industry, and we have a small incubation center where we incubate people. In 21, we started a PhD mini symposia. It's an in-house activity just to have a scientific science feast within our own niche. And it's a very sustainable model. And we finished the fourth symposia on 6th and 7th. So the plan is that 6th and 7th we host the symposia. 7th we host in a memory of Professor Bhan. And that day we do recognize our people, which we will be doing today. And former directors and former secretaries and all the NIBNs, I must put a word of salutations. This is all we are. And we are continuing. Precisely, we are 13 years old. We just entered to 13th year. This is what our scientists have been doing. And these are the technologies which have been either transferred or are under pipeline or under commercialization. 4 of our kids are waiting for Mantriji's we're waiting for a date so we can move ahead for commercialization. And today is a day when Dr. Girish's lab has got a patent. It's a second patent in the year. So we are very happy for Girish, you and your team. It was 16th of December when the paper came, and he got it this morning. So you made this day more special, you and your team, through the work. It was 16th of February, again the same year, when another zoonotic disease, parasitic infestation, Toxoplasma gondii, Abhijit got. So year 2024 could give 2 patents to this 13-year-old institute, and we are moving ahead. So vaccine and diagnostics remains our aim majorly for that. Indigo chip is a signature work for this institute. And another first national One Health Consortia, which was started by DBT in 2021, it's coming, moving very well, and is coming to its conclusion. Next year, we put our conclusive report and summary on table that what did we do. zoonotic diseases. I won't go into the detail. We have people working on leptospira, JV, TB, and as I said, brucella and toxo, besides other things, and rabies, of course, a bit of work. So with that, this was a flagship project, and there were good leads for both bacterial as well as viral diseases. So group which is led by Dr. Syed Faisal is moving ahead with a bunch of vaccine candidates. For JEV, we could get the one LFA, and it has been transferred to a company. Then we work on control and prevention measures. As I already said, and then we move on with the last 2 years, some new scientists who have joined us. So we make different organoids. So the work started with the making placental organoids, and brucella is a pathogen which they have taken up. So this is moving. As I said, tuberculosis, so this was one work. So BRIC has already launched one genome one day. Last week, our genome was depicted, and this is the first complete genome by the team led by Dr. Bapaditya Day. And this was a complete genome of the Mycobacterium murigis. We're all very much concerned about it. His laboratory could also make a 3D pulmosphere with which they are moving ahead. And this is a story of toxoplasma. I'll reframe going more into the details, but it's very important diseases because for some it might be a low hanging fruit. However, I see it this way: leptospira and toxo, they are zoonotic. But when the human patient reach to doctors, majority of the time it's pretty late. So there is a attention to be given, and this is the only institute, and the groups fortunately are working very well. There is a reproductive biotechnology group which has developed a PICAS model in case of K-prime model, which could really be taken for the human. Animal bioreactors, as I said, Dr. Nirmalya Ganguly's lab is there, and he could really come out getting FSH through the milk of this transgenic animal, and it has been tested, and we are looking forward for expanding it further, going ahead and looking for a industry partner. And now he's continuing with the goat model to generate a therapeutic proteins further. Then coming on to we also work on a regenerative medicine, so therapeutics, so that's what we do. So this is a group, people work on adipose generated and marrow generated and fetal origin generated stem cells, use a lot of different kind of scaffolds. We are material scientists and organoid people, so we do that. So 2 years back, fortunately, we have a nutrition scientist, so we are enriching this area at the institute, which is there. Institute has a state-of-art facility for small animals. So we now have the ABSL2 at place from 23 onwards. It's commissioned and is in use. We have a ABSL3 laboratory currently is on sharing way, but we look forward to have it over here. This is a vaccine testing facility. Dr. Hegde had to go for a while because state government is very seriously pursuing it, and we are also wanting to use it. With that, thank you so much. With that little, hope I could, express it to our esteemed speakers a little bit, what exactly NIAB is marching ahead with. Thank you, and we look forward.
Thank you very much, madam. So recapitulation of our journey is definitely a motivating thing for all of us, especially for those who are there from the beginning. Professor Redana could appreciate that. I now welcome our distinguished speaker, Dr. Sudhanshu Vrati, sir. Dr. Vrati is a renowned virologist. He served as Executive Director of the RCB, Regional Center for Biotechnology in Faridabad, India, where he played a very transformative role in shaping the institute into becoming a premier center for advanced research, innovation, and education in life sciences. He earned his PhD from the Australian National University, Canberra, his DIT in biochemical engineering from IIT Delhi, his postdoctoral research at CSIRO, Sydney, and was also visiting scientist at Pasteur Institute, Paris. Dr. Rati's research was focused on understanding the molecular mechanisms of viral replication and pathogenesis, and he's more interested in developing vaccine platforms and therapeutics, and has made a significant contribution to the development of all these, which I mentioned, for various viral diseases. Throughout his illustrious career, Dr. Rati has received numerous awards and honors for his outstanding contributions to science. I would like to specifically mention he is the fellow of the Indian National Science Academy, Indian Academy of Sciences, and the National Academy of Sciences, India. It is really a privilege to have you here today, sir. And we are looking forward to hearing to you on the topic making of the Indian rotavirus vaccine. Please join me in welcoming Dr. Rati.
You have a pointer? Yes. But what about the pointer? Your pointer?
This is long press for pointer, sir. Not sure. Slide and paste.
I think I'll just use it overhead. And where's the mouse? Okay, good afternoon, everybody, and many thanks for very, very generous introduction. Thank you, Dr. Tharu, for this invitation to speak on this, Dr. M.K. Bhan Memorial Lecture. I will, in 2 or 3 sentences, try to describe Dr. Bhan, and then very briefly, I will describe how, I came about interacting with him. And after that, I will describe some work that we did together on development of India's first rotavirus vaccine. Dr. Bhan was an extraordinary individual. And as a human being, I think he was very humble and very down to earth. For a young researcher to be able to talk to him, he would make it possible by making them comfortable all the time. And as a scientist, he was extraordinary clinical research scientist and a great visionary. At Department of Biotechnology, he can be credited with establishing the most number of autonomous institutions. So he was institution builder, and one such institute that he created is this. There are several others, including THSTI and RCB and others. My interaction with Dr. Bhan started in 2005 when I think he moved to DBT around that time. And I had met him only once at Bangalore airport, actually. We were giving a lecture together at IISc. And after that, we were taking the same flight. And as Dr. Bhan, I did not know him. So he said, let's have beer together. So we had beer together, and then we just, flew to Delhi. He went to AIIMS, I went to NIA, and that was it. But in 2005, I got a phone call from Dr. Bindu Dey. She was the advisor looking after infectious diseases at that time at DBT. And she said, Secretary Dr. Bhan wants to talk to you. I said, sure. And so he told me, after, standard greeting things, etc. He says, Sudhanshu, I have now moved to, AIIMS. From AIIMS, I have moved to DBT. And, I want your help in this vaccine project. And I said, but I have no knowledge of making vaccines or doing anything on vaccines. He says, I don't have it either. So, but we will learn together. Let's do it together. And I think that excitement was enough for me to get on board. And I will tell you then the whole story of how this rotavirus vaccine came about. So, I got involved when the vaccine clinical trial started in India. I will tell you the story. Vaccine clinical trial started in US, then it moved to India, and I got involved at that point of time. And my responsibility was to develop a group who would establish validated assays for vaccine efficacy, for virus identification, etc., related to vaccine efficacy. So this required GLP, good laboratory practices. And I was working at NII, and NII being a standard research institution, there was no concept of GLP. Students are wandering here and there, they're stealing each other's buffers and solutions and whatnot. So at that point of time, I told Dr. Bhan, I said, I have no experience of GLP, but what I have read about GLP, I don't think this is possible at NII. And he says, एक नया institute बनाते हैं. And that was the genesis of THSTI. You know, the first major project that THSTI undertook was clinical development of rotavirus vaccine. So you can imagine, you know, he was such a great visionary that if something has to be done, he would make it possible. So because he realized that, you know, NII would not be able to undertake a GLP-based clinical evaluation of a vaccine development, he said, let's make a new institute. And THSTI started, then RCB started, NIAB started, so many institutions started. Okay, so I will be talking about this rotavirus vaccine story, the Indian story, and this is an extraordinary Indian success story where an idea or an observation by Dr. Bhan in his clinic led to development of a vaccine that has gone into clinic and has been extensively used in the field. Now, this is a very good example of translational research. And let me tell you what translational research is all about. This is a cartoon at the top that I have taken from Nature. This is a cartoon showing Valley of Death. And Valley of Death is a valley in California where apparently it has not been easy to, you know, travel through that valley. The temperature goes up to like 56 degrees centigrade. And many people died trying to cross that valley. It is exactly the same happening in science and the clinic. On one side of this valley, we have a scientist who has some idea, who has some you know, observation in the lab, which can give some benefits to a patient, which is sitting on the other side of the valley. A patient is hoping that scientist will come up with a solution of his problem. Scientist is hoping that he will have a solution for the patient. But bridging the distance between scientist and patient has not been easy. This is like crossing the valley of death. Lot of our ideas or discoveries that are made in the laboratory, they die without being converted into a meaningful product. So, in 15 years ago now, or almost 20 years ago, this new kind of science started to be talked about, that is translational medicine. And in a crudest sense, translational medicine is converting a discovery or an idea generated in a laboratory to a product or process that could benefit the patients at the other end. But this is a very complex process. You know, it is very expensive. It takes a lot of time. It requires expertise of various different kinds, like systems biology, cross-species validation, bioinformatics, innovation, markets, science politics, etc., etc. And therefore, many of us who are in our laboratory are neither equipped to deal with all of these together, nor we understand the intricacies of this translational medicine. So therefore, now experts and facilities are coming up, which undertake, you know, discoveries from research labs. And since they have these expertise of converting these ideas into product, they undertake the translational research. Now, translational research is also very risky in terms of success. I'm quoting this paper from American Journal of Medicine, where the authors cataloged the research reported between 1979 and 1983, and they found that there were 101 discoveries which were claimed during this time to have a medical significance. But by 2002, only 5 of these discoveries had had received the license for clinical use. So chance of success are very, very low. You can see about 5% ideas really get converted into meaningful product. And it's very long time that it takes. From the same report, I found that the time it takes for an idea to be converted into a product ranges anywhere between 14 to 44 years, the median being 24 years. So majority of our scientists have active life of about 35 scientific years. So therefore, there's a very little chance that they will be able to go through all of this journey in their lifetime. But here I will present a extraordinary scenario where all of this happened in one's lifetime. Now, vaccine development timeline is long, or has been long. I mean, COVID has made us learn how to do these things faster. But again, this has been taking anywhere about 15 to 20 years in the past. So once you have an idea, you've identified an antigen that could be useful in your laboratory, you have to do extensive preclinical testing. You have to do the toxicology, etc. And with having shown the immunogenicity in the animal model, as well as protective efficacy and its safety in the animal models, then you are given regulatory permission to test in the humans. And that starts with phase 1 clinical trial, which is essentially a safety trial in a small number of individuals, anywhere between 20 to 100. Once you establish the safety of your product, you are given an opportunity to conduct phase 2 trial, where you can test more than one dose, and you can test the immunogenicity, etc., along with safety. Having demonstrated the safety and immunogenicity of your product, then you are eligible to apply for phase 3 trial, which is done in larger number of individuals because you need to demonstrate the efficacy. And the whole process takes anywhere between 15 to 20 years. After demonstrating the efficacy and safety of your vaccine after phase 3 trial, if you are lucky, you will get regulatory permission to market the product. But story doesn't end there. You still have to, your vaccine or your product is still, under watch, which we call phase 4 for long-term safety issues. So development of vaccine is a long process. It takes anywhere between 15 to 20 years. It's risky. Few ideas become products. It's complex, requires multiple expertise, team of experts, etc., as I said, for any other translational product. So vaccine development is also a team game, and it requires, molecular biologists, scientists, biotechnologists, product development specialists, clinicians, statisticians, public health specialists, population biologists, and social scientists. And I have just listed few key people in this team. On the left, you will see Dr. Bhan and Dr. Durga Rao from IIC Bangalore. These were the people who identified viruses that could potentially be made into vaccines. We had, as I said, that I was leading a team to develop laboratory assays for vaccine efficacy and safety. Then we had 3 key people, Dr. Neeta Bhandari from New Delhi, Gagandeep Kang from CMC Vellore, and Ashish Bhavdekar, KEM Hospital, Pune. These were the people who were carrying out the clinical trials of the vaccine. And we had international experts, Roger Glass and Harry Greenberg, and several others who supported us through Indo-US Vaccine Action Program in developing this vaccine. So let me tell you a little bit about rotavirus. Rotavirus was first discovered in humans in 1973 by Ruth Bishop and her colleagues in Australia. The virus is a wheel-shaped virus, which was seen under electron microscope by Ruth Bishop and her team in infants which were suffering with severe gastroenteritis. This is a non-enveloped virus, which is triple-layered virus with a icosahedron capsid, 75 nanometer in diameter. There are 5 serogroups of rotavirus, A, B, C, D, and E. Humans are infected primarily by serogroups A, B, and C. All 5 serogroups cause disease in animals as well. And several animals, and almost all animals get infected with one or the other rotavirus. Now, interestingly, the rotavirus genome is segmented. It has 11 double-stranded RNAs that code for 12 proteins. And as I said, these viruses can infect humans as well as animals. So in animals, basically if a human virus is infecting and if that animal happens to be infected with animal virus, and since the genome is segmented, there's every possibility that there will be reassortment of genome and recombinant viruses would be generated, human animal reassortant viruses would be generated. And there are a lot of such examples of such reassortments. There are rotavirus serotypes which are defined by two key proteins, VP4 protein, which is involved in virus entry. It is found on the virion surface, and VP7, which is another immunogenic glycoprotein, and it has the protease. So VP4 has the protease activity, whereas VP7 is a glycoprotein, which has several other functions, including the virus uptake and interactions. Now, VP7, there are several different genotypes of VP7. There are 16 of them described so far, and they are called as G1, G2, G3, G4, G8, etc., etc. VP4, there are 28 different genotypes of VP4, and just example is P4, P6, P8, P9, etc. So these viruses are defined at the genotypic level by the identity of their VP4 and VP7 genes, and a combination of G and P is how these rotaviruses are described. Theoretically, almost 500 different kind of genotypes of rotavirus can exist, because combination of VP7 and VP4 would produce almost about 500 different genotypes. But the most common genotype is G1P8, which is almost like 50% of the circulating genotypes, and the others are G4, P8, etc. So rotavirus is transmitted to humans by fecal-oral route. Rotavirus enters the our digestive system and reaches the small intestine, where the virus binds to the cells at the villi, and virus infection causes atrophy of these cells. Once the cells are sick or die, the capacity of these cells to absorb carbohydrate and liquids gets compromised, and that leads to diarrhea. Now, rotavirus diarrhea is the leading cause of severe gastroenteritis in children, young children less than 5 years of age. Almost 30% of all severe acute gastroenteritis cases are due to rotavirus infection. Approximately 600,000 global child deaths are attributable to diarrhea. Of this, as I said, that 30% are because of rotavirus, so about 200,000 deaths every year are because of rotavirus. This was the scenario, when this vaccine research was being undertaken. You can see, each dot on this map represents about 2,000 deaths each year, 2,000 kids dying each year. India had a load of about 400,000 hospitalization due to rotavirus infection, about 100,000 deaths each year. That means one death due to rotaviral diarrhea for every 250 children born. Rotavirus disease cannot be treated with antibiotics, and there are no antiviral drugs available to treat rotaviral infections. Most children are at risk of infection. Therefore, vaccination offers the best hope for preventing severe rotavirus illness. The rotavirus vaccine development has taken the Jennerian approach, and we all know in the Jennerian approach, a live attenuated virus, be it human or animal or reassortant between human and animal, has been used for developing a vaccine. The same way, the first rotavirus vaccine was developed in 1988 and licensed in US. The vaccine was developed by Wyeth, and the brand name was RotaShield. This was a human simian rotavirus reassortant tetravalent vaccine. But after having gone through phase 3 trial and vaccine being launched into the market, the vaccine had to be withdrawn, because during the phase 4 trial, it was found, the post-marketing survey showed that vaccine had association with intussusception. Now, intussusception is basically telescopic folding of the small intestine that can cause edema, perforation, and that can become life-threatening. The number of interception cases was about 10,000 vaccinated infants, one interception per 10,000 vaccinated infants. Now, the phase 3 trial for, RotaShield was done in numbers smaller than 10,000. And therefore, this, interception thing was missed out. So because of this interception, the vaccine was considered unsafe. And, as I said that during the post-marketing surveillance, also if the vaccine is found unsafe, it can be withdrawn, and RotaShield was withdrawn, having successfully completed phase 3 trial. But in post-marketing surveillance, the vaccine was found not safe. Then in 2006, 2 new vaccines were reported. Phase 3 trials were reported in New England Journal of Medicine. One was Rotarix by GSK. It was a monovalent vaccine which was based on G1P8 strain of human virus. It required 2 doses of vaccine given orally. Rotatech was reported by Merck. It was a pentavalent vaccine which was made on bovine virus backbone. Okay, and it required 3 doses of vaccine given orally. This time, to rule out any issues related to interception, these vaccines were tested in 60 to 70,000 kids at phase 3 trial. And it was found that there was no increased risk of interception and there was no severe adverse events associated with vaccination. The efficacy of vaccines was found to close to 90 to 98% against severe disease. And based on the efficacy of these vaccines, WHO made a recommendation of use of these vaccines in national immunization program across the world. But when these vaccines were tested in low and middle income countries, in let's say Vietnam or Africa, the efficacy was found to be low, anywhere between 50 to 60%. But that was not very surprising because other oral vaccines had a similar history in developing world. For example, oral polio vaccine that we all know is given repeatedly to ensure high immunogenicity and efficacy of this vaccine in India and other countries. Oral cholera vaccine had the same issue. It was found to be less immunogenic than in other countries when it was tested in Thai and Indonesian settings. Now, what might be the reason of low immunogenicity of oral vaccines in low and middle income countries? In Asian and African countries, mothers have the tendency to breastfeed their infants as soon as they start crying. And no matter what you do, when you give oral vaccine or anything to these infants, they cry, and mothers have the tendency to breastfeed them. Now, it was thought that mother's milk contains very high levels of antibodies and very high levels of antibodies that neutralize rotavirus. So it was thought that it is possible that these vaccines virus are getting neutralized by mother's milk, and therefore is leading to a lower efficacy of the vaccine. And there are several other reasons, including malnutrition and continued infections of gut in infants and LMICs. So the hypothesis was, if a vaccine is made from an Indian virus that establishes well under the conditions of breastfeeding, can we make a more effective vaccine? And the story starts in 1985 at All India Institute of Medical Sciences, where Dr. Bhan used to run a pediatric clinic. He identified that there was an outbreak of asymptomatic rotavirus infection in newborn unit. So asymptomatic means the kids were happy, but they were doing the surveillance for rotavirus, and they found that a lot of kids which were born there, their stools contained rotavirus. What his team found that 50% of the infants got infected within 3 days, and 75% got infected within a week of living in that newborn unit. He identified the virus as 116E virus. This was the name given. And the virus was identified as G9P11 genotype. This was a human bovine reassortant virus. His team continued to follow these kids which were infected with the rotavirus during their, in the nursery for next 2 years. And he found that these kids who got infected with 116E had fewer and milder episodes of rotavirus diarrhea. A similar observation was made in Bangalore by a team of Dr. Durga Rao, and he identified a virus called I321, which was different from 116C. And this was a bovine human reassortant virus. The 116C was human bovine reassortant. This was bovine human reassortant virus with G10P11 genotype. A similar follow-up for 2 years showed that kids who got infected with this virus had fewer episodes of rotavirus diarrhea. Now, this slide shows you the genetic composition of the viruses that were used by GSK vaccines and Rotatech vaccine. While the Rotatech vaccine on the right, everything is red. That means majority of the genes are of the animal virus origin, except the black one, which is VP7 gene. GSK was all human virus. Everything is black. 116C was all black except for VP4 gene, which is the surface protein. And I321 is majority of genes are bovine origin, except in non-structural proteins, NSP1 and the VP6. And NSP3. So these were non-structural proteins of human origin there. So since both strains colonized the infant gut extremely well, it was thought that maybe the maternal antibody was not neutralizing activity, was not neutralizing them because the bovine outer capsid, it was not human, VP4. And they caused no disease in kids. And the virus was shared almost for about a week, indicating that there was a good take of the virus. And both infections were associated with protection against disease or reinfection. So this, made people think that these could be two good potential